Buenos días. Hello to everyone. Good morning, Adam. For us, it's a pleasure to welcome all of you to this talk that is going to be given by Professor EDVARD MOSER, a Nobel Prize in 2014 for his discoveries, not alone, because science is more and more a collective enterprise concerning what is called the GPS, the human GPS, so how we really identify where we are, something that is really very important. So thank you very much, Professor Moser, for being here. I want to thank the Nobel Foundation for giving us this opportunity of hosting this talk. And I think it's very important, just two things. I have 2 minutes, and I am only just using these 2 minutes. One, and as I said before, science is a collective enterprise. That means that we have to collaborate different people and also from different disciplines. And this is a really magnificent example of this. The background of Professor Moser starts with some mathematics, statistics, programming, psychology, neurology, and big problems have to be tackled with the help of different disciplines and different backgrounds and different point of view. And that's the only way to do that. So thank you very much for being here. It's a pleasure. Thanks to the Nobel Foundation for making this possible, and we hope that this is the first occasion of something that is going to be repeated more and more. Thank you very much. Now, please.
Do I turn it on? Okay. So, hello everybody. I'm Adam Smith from Nobel Media, which is the media arm of the Nobel Foundation, based in Stockholm, Sweden. And on behalf of everyone at Nobel Media, I'd like to welcome you to this morning's lecture. We are here in Madrid because yesterday we organized a Nobel Prize dialogue on the future of aging with the Fundación Ramón Areces. And Edvard was one of our Nobel laureates who took part in that discussion, a very intense day of discussions. And I guess Edvard is probably quite tired now because it lasted all day long. But as part of those dialogues, which we run all over the world, we like to organize events such as these, where we bring the Nobel laureate to meet especially the next generation of scientists and spend some time getting close to them. And we're really delighted to be here. And I'd like to thank Complutense very, very much for the kind invitation to join us. Thank you very much. I'd also like to thank Edvard for giving us an extra day of his time. He's one of those Nobel laureates who's quite difficult to prize out of the lab. Some of them like to travel a lot, but he likes to be in his lab, so it's good to have him here today. I encourage you to listen very carefully to his lecture and also to ask questions in the Q&A afterwards. And I don't want to take up any more time. I just want to say enjoy the lecture and welcome. Thank you.
Thank you. So the Dean of the Medicine School, and I just want to thank him especially also before he has his 2 minutes, to thank him especially for hosting us here in this building. Javier.
Thank you very much. Just on my part, just also to say welcome. Thank you for coming. It is a great honor for us to have you here. This is an very old faculty. We have had the honor of give to the society another Nobel Prize, Santiago Ramón y Cajal, but it was in 1906, very long ago, too long ago. Now we are committed to motivate our students to try to, that's because it is very important for us to be here, because we would like to convey a message for the students. Maybe quoting Obama, yes, we can. Thank you.
So, thank you very much, Professor Moser. The floor is yours.
Good morning, everyone. Thank you to the rector and to the Now? Or too much? Well, I can't do anything about... Oh, now it's better, right? It's better? Yeah. Okay. Yeah. So thanks again for the invitation, and I'm happy to be here to speak to see so many young students. Hopefully this can inspire someone to choose to go to neuroscience. What I'll do today is to discuss with you some of the higher brain functions where I will emphasize space and time. So especially space, which is the area where I've done most of my research. But why study space? Why study our navigation system, as we often call it, or how we find out where we are, how we get from one place to the other? So these are very obvious functions that we have, and very often we don't really think about them, but it is totally critical for survival that we actually can get from one place to the other and know where we are. Otherwise we will be killed immediately. So but the reason for studying space is not only because it is an important behavioral function, it's also because it is one of the higher brain functions that is comparatively easy to study, because for several reasons. It can be studied in rats and mice. They are very good at it, and as you will hear later, the systems in the brain for navigation are quite similar in rats and mice, which we study, and in humans. And it's also quite easy to study space in the lab, in experiments. You can design tasks where you ask a rat or a mouse to find its way through a maze, for example, and you can measure it very easily. So it's much easier to study space, for example, than, let us say, abstract thinking or language or planning or imagination. Those things are much harder. So it's a good place to begin, and there has been quite a lot of progress in this area over the last few decades. So where is space located in the brain? Well, first of all, I mean, it's not really one area. I mean, the whole brain is in some way involved when you try to find your way from one area to the other. But yet there are some areas that are particularly important, and two of them are shown here. So one is the hippocampus, which is the red structure. It is part of the cortex, but it's actually on the inside. So that doesn't quite come across of this image, but is where the medial temporal lobe folds around, and then on the inner side, but still an old part of cortex. Hippocampus is strongly connected to entorhinal cortex, and most of its connections to the rest of the cortex go through the entorhinal cortex. So entorhinal cortex both provides inputs to the hippocampus and also receives many of its outputs before it goes to all over the rest of the cortex. So those two areas are critically linked, and I'll talk about them. But the challenge is that even if we do want to ultimately understand the human brain, or at least also understand the human brain, it is very difficult to access, and because the processes we want to observe are very, very small. So things happen distributed among approximately 10 to the 11th of neurons and 10 to the 15th of synapses. There's so many of them in the brain, and they're so small and they're so distributed that it is an immense task to try to find out what's going on in any brain function. But it is easier to do this in smaller organisms, and neuroscientists study a number of species. So rats and mice are probably the most commonly studied one, but not the only one. A lot can be done in much simpler species like flies and even the worm, the nematode C. elegans, with its 302 neurons can be useful in some circumstances. But if we want to preserve some relationship with humans, then we often end up in the middle, and the middle is very often the mouse. So this just shows the hippocampus in 3 different species. So you have humans to the right, and you again see the same picture, and at the bottom you see a transection through the hippocampus with its sub-areas that are include the CA1, CA2, CA3, CA4, and the dentate gyrus. In the middle you see a monkey, not too different, and to the left you see a rat hippocampus. So the red thing again, and the entorhinal cortex in blue. So differently located in the brain, somewhat differently because it's folded and organized in a different way, but actually internally it consists of the same substructures. So again, dentate gyrus, and then the CA3 and the CA2 and the CA1, and they are connected in the same way, consist of the same types of cells with the same properties, and as you will see, many of the functions are the same as well. So there's a lot that can be learned by studying the rat or mouse hippocampus, and much of the history of the area of space actually is from the rat, and now more recently the mouse. So much of the history of this field can be traced back to Edward Tolman. So this is almost 100 years ago. So in the beginning in the 1920s and then extending up to the 1950s, Tolman was a psychologist, experimental psychologist, who worked at University of California at Berkeley. And what he proposed was that our brains have internal maps. We use maps when we navigate, and we use maps when we organized our knowledge. And this was a radical change at that time to the rest of what was called behaviorism. Behaviorism was a tradition of experimental psychology where investigators tried to understand behavior by breaking it down into small chunks. I called it stimuli, which were the influences from the world, and then responses, which were the behaviors, and the responses were broken down into small pieces, and then they tried to understand how sequences of responses happened and how they were dependent on interactions with the world. But this sequence view did not fit with Tolman's view, because he said it's more than just sequences. It's really an organized picture of the world that we have in our brain. And he based this on a number of experiments, and one of them shown here. I won't go into the details, but what this suggested briefly is that rats are able to take shortcuts and detours, and when they find their way. And the ability to take a shortcut is important, because if a rat can take a shortcut, it means that it finds a way according to knowledge about how places in the world fit together. If you only found your way by learning along sequences of A, B, C, D, and E responses, then you only strengthened what you had done before. But if you take a novel shortcut, one that you had never walked before, that means that you must have some knowledge about how the world is organized in a map-like way. And this was very controversial and remained controversial for a long, long time. And much of the reason was that Tolman or no one else had the techniques to go into the brain and find out if there are such maps in the brain. No one knew, really. He just inferred them. So it was only in almost 30 years later then John O'Keefe used microelectrodes, very small electrodes that at the tip could pick up the electrical signals of neurons to find out what's going on in the brain when an animal, and that an animal was a rat, did for different things. So in this rat walked around in a box, or it could be in a maze, and he was then able to put these small electrodes into the brain. They're very, very small. They're about, nowadays, usually around 15 to 20 micrometers wide. So small, they're actually smaller than the cells. So you can squeeze them in between the cells, and it picks up the natural electrical activity of the cells without damaging them. So these were then fixed to the skull, and then when the experiment went on, the rat walked around in the box, for example, and he could then pick up the electrical signals and see if there was any relationship to what the rat was doing. So in the next slide, I'm going to show what he found. So this movie will show a rat. You see it from above, from the ceiling, and the rat is going to walk in a box, and this box is 1 meter by 1 meter, and the rat will walk around to collect small pieces of chocolate that are lying in the box, and that's enough. And that's just a trick to have the rat running around, really, to visit every possible place. And while the rat is running around, then in this experiment, we were recording activity of one cell in the hippocampus, the red area, of the rat. And then each time you hear a sound, this cell is electrically active. That means it's firing an action potential. You will also see a red dot appearing on the screen each time the cell is active. So if I now start this movie, so this is a cell firing, as we call it. That means that you hear action potentials of one particular cell. And the rat is, we speed it up a little bit, but it's running around to pick up its chocolate pieces. And I think what you already see now is that this particular cell in the hippocampus is active only when the rat is in the upper left part of the box. And so this is very typical of hippocampal cells. O'Keefe called them place cells. And he called them place cells because they are active at one place. And the area that contains activity is called a place field. So it turned out that many, or actually even perhaps most of the cells in the hippocampus are like that. They're active only in particular regions. So while one cell may be active, like shown here in this heat map, red is high activity and blue is low activity, is active only in the upper left, and other cells might be active in the middle, and yet other cells might be active in the bottom left or bottom right, and so on. And imagine then that you have 100,000 of such cells in the hippocampus. That means that at every possible place in this box, there are cells that are active, and it's a unique combination of cells that is active at every possible place. And that in turn means that if you now, let's say you could access all of these cells, then, and you fed that activity from that combination of cells into a computer, and then ask the computer, where is the rat now? The computer could tell immediately the rat is there and not there, because the combination of active cells is different everywhere. So that then led O'Keefe and Nadel, his colleague Lynn Nadel, to suggest in 1978 that this is actually a part of the map that Tolman had suggested 30 years earlier. So we have an internal map in the brain, and this map consists, according to their hypothesis, consists of place cells that map the environment, so that when the brain, when a downstream area of the brain sees a combination of active cells, it can tell where the rat is. So in that sense, it is a map. So this was in 1971, and then years passed, and another big event happened in the late 1980s. So in the late 1980s, there was another cell type that was discovered by Jim Rank and his student Jeff Taube. They found out that in a different brain area, not in the hippocampus, but somewhat outside, in an area called the presubiculum, there are cells that respond to the direction of the animal. So when the rat is walking around in the box, these cells fire, for example, only when it's going to the left, or only to the right in the box, or only upwards or down. So What you see at the bottom left there is a plot for one cell that is a so-called head direction cell. It shows the firing rate, the activity of the cell as a function of the direction of the animal's movement or the animal's head. And as you can see, it fires specifically only when the rat is walking in about a direction of between 240 and 300 degrees in this case. So that's late 1980s. And then another 10, 15 years passed. And this is when Britt Moser and I started a lab in Trondheim. And the question we started out with is then, how do these place cells of O'Keefe actually arise? And how are signals from place cells integrated with signals from head direction cells? How does it come together? How do they work together? So we started our lab in 1996 with that in mind. And the first breakthrough came in 2004 when we had decided that we started to record activity from the medial entorhinal cortex. So that is the blue area that you remember from the human brain I showed. So this is in a rat brain, and the rat brain is seen from behind, and you see in what corresponds to the medial temporal lobe, you see the colored area, that is the entorhinal cortex. And in the upper part of the entorhinal cortex, that's where we recorded activity. And the reason for doing that is that this area provides the strongest input to the area of hippocampus that has the place cells. But it also, because it links, this is a place where head direction signals come in and go to the hippocampus. So it might be an interface area that could potentially link directional signals with place signals. So what we found then, 2005, was that this brain area does contain cells that respond to position, like the place cells, but is also somewhat different. And what you see here is that this is, if you look at the right first, this is a picture of a box, activity in a box again. So the box is seen from above. This box is a little bit bigger than the one you saw. It's 2.2 by 2.2 meters. And the gray trace in the background is the path that the rat walked over the course of 30 minutes. And each dot here is like the red dot that you saw on the movies. So what you can see is that the red dots or the black dots, they form clusters of activity, many clusters, not just one like in the place cells, but it's many of them. And those clusters of activity form a very regular pattern that repeats itself all over the space. So a triangular pattern, you can see, or a hexagonal pattern, to be more precise. And because it forms this hexagonal pattern, it is like a grid that covers the entire space. And for that reason, we call these cells grid cells. And then it turned out there are many types of grid cells in this brain area. So anywhere where you record using these electrodes, so here they are magnified really. So you, as I said, each of them are about 15 to 17 micrometers wide. And you pick up the electricity from the bottom of this. And here are the cells underneath that give rise to that activity that you pick up. And then 3 simultaneously recorded cells. They are illustrated here in blue, red, and green. They have patterns like this. So you see there's a green grid cell, there's a red grid cell, and there's a blue grid cell. And what you can notice is that they cover different places in the space, different XY positions in the environment. So together, these already cover quite a lot of the environment. So if you then imagine you have several hundred thousands of these, they cover every possible place. So, but the organization is pretty chaotic in the sense that neighboring cells have different phases, as we call them, different XY positions. So it's often called a salt and pepper organization because it's just randomly mixed. But then, while the phase is randomly mixed, that's not the case for the scale of the grid, because the grid cells exist at different scales. So you have the ones with the smallest scale. They're typically located in the upper or the dorsal part of the entorhinal cortex. So entorhinal cortex is the red area here, and this is a sagittal section through the brain. This is the hippocampus. So red area, hippocampus, and the upper part usually has a smaller scale. That means that the frequency of the grid is quite high. The distance between the fields is very small. And then this scale increases and gets wider and wider as you get further down into the brain. And at the end of the brain, it is very large, perhaps several meters between each of the grid clusters. And that's just shown here. As you go down, then the scale increases. So that then raises the question, is this grid actually continuous, that you begin with the smallest one and then it gets wider and wider and wider as you go down? Or are there separate maps? So maybe it could be three or even more different maps. So there's a small map at the top, and then there's an intermediate map in the middle, and then a larger map further down, and maybe further. So in other words, it's a continuous map or it's a discrete step-like map. And it turns out to be the latter. So this is a plot from one single animal. So you can see that when you have enough cells from the same animal. So what you see here on the x-axis is the position along the length axis of the entorhinal cortex. So left is dorsal or top, right is ventral or bottom. And then on the y-axis, you have the size of the grid. That means the distance between each of the points. And then each dot is one cell. And what you can see is that as you start from the top, the scale is small and gets bigger and bigger and bigger, as we knew. But you can also see that it's actually a step-like increase. There's nothing in between the steps. So here there are 4 steps, and we often call those modules. So module 1, module 2, module 3, and module 4. And these modules even have actually a specific scale relationship, in the sense that as you go from module 1 to module 2 and module 2 to 3, there's a fixed relationship, so that the scale of module 1 and then you go to 2 is about 1.4 times as large as number 1. 3 is about 1.4 times as large as number 2. And 4 is about 1.4 times as large as number 3. So this is like a geometric organization, or a geometric progression. So you multiply by the same factor to get from one level to the next. And that turns out then to be a very efficient way to organize these maps if you want to cover the environment with high precision and still use as few cells as possible. So when we found these cells, it was very often presented as the GPS of the brain, and that is the popular way to describe these cells. So there are, of course, similarities and there are many differences, but the similarity is that these cells depend a lot on the animal's movement in the space. So when the rat is walking around, the brain counts, let's say, counts the steps, and counts the distance that the animal has moved, and also the direction, so that by keeping track of this movement, it can get the cells to fire at very, very regular positions, regardless of how it moves between them. So this is what the GPS does as well. It tracks the movement, distance and direction, and doesn't really care about what the environment looks like. And the same here, these grid cells keep their patterns depending on your movement, but they don't really care about what the room looks like and what you see in the background. So in that sense, it's similar to a GPS, but of course it's much more complicated, because these cells interact with all kinds of other information that corrects it and makes it much more precise. So it turned out then that these grid cells coexist with a number of other cells. So I mentioned the head direction cells, those that fire only when the rat is walking in a certain direction or keeps its head in a certain direction. So these are shown here. They exist in the same area. This is again entorhinal cortex, medial entorhinal cortex. And you can see they don't have any hexagonal pattern. They just fire essentially everywhere. And what you see to the right is a plot of activity as a function of the direction of the rat. And you can see this cell only is active when the rat is going towards the left. Same here. This cell is only active when the rat is going towards the left and upwards, and same again here. So these are direction-sensitive cells. And then there are other cells that respond to the speed of the rat as it's walking around in this box. So you see that in the plot to the left here. This is 7 different cells, each shown in a different color. And in the background in gray, you have the speed of the rat as it's walking around in the box. And if it's perhaps easiest to see if you focus on the yellow one. So yellow is the activity of that particular cell, and gray is the speed of the rat as it's walking around. And you can see how closely the two curves follow each other. So that means that these cells actually express the speed of the rat as it's walking around. It's almost like a speedometer. So in other words, we have information about direction and speed, just like what a GPS needs. That is already present in specialized cells in the entorhinal cortex. And again, these cells don't really have a preference for any particular location in the box. They're active more or less everywhere. So that turned out then not to be the only type of cell that is present, because a few years later we found out that there is a distinct type of cell that rather responds to the boundaries of the environment. So the boundaries in this case, when the rat is walking in a box, is the walls. It could also, when it's walking on a table, it could be the edge of the table. So you can see that cell here only fires when the rat is on the east side of the box. And that happens even if you stretch the box in one direction or in the other direction. Or if you then go to a different room, it chooses, in this case, the west side instead. And if you insert a wall, it even fires along that wall as well. So these cells are completely different from the grid cells, but they indicate the local boundaries of the environment, which is also important to know about if you are a rat and want to navigate your way in that environment. So this is the situation a few years ago. You may then wonder, are these cells only present in rats and mice? Well, the answer is no. So they were all found in rats, and turned out later that mice also have them, not very surprisingly, because this shows an evolutionary tree for mammals, and rats and mice are of course on the same branch for rodents. But then a few years later, they were found in different group in bats. So bats are not small rats or not small mice. They are on a completely different branch, and yet they have grid cells. And they were found in monkeys, and they were found more recently in humans. And you may wonder, how do you record grid cells in humans? Well, that's can be done in a few cases where patients with very serious epilepsy that cannot be treated by drugs will have electrodes implanted in the brain to monitor tissue and find out where the seizure starts, which you want to do before you decide about doing a surgery and where to do the surgery. So this is in serious cases. But in those cases, you get for free the activity of the cells, because these patients have to walk around with electrodes maybe for 2 weeks before the surgery. And then these data can be collected, and turns out that also here you have grid cells. So both the grid cells and the head direction cells and the border cells seem to be present across all, or at least most, or probably all mammalian species, or at least in very many of them. So it probably arose quite early in evolution, and then I think there are many reasons to believe that what we find out here in the rats and the mice is probably also taking place in the human brain. So, well, now we say there are many cell types, but it actually turned out not to be the end. So I want to just show you briefly data from another experiment where we, together with Øyvind Højdal and 2 of his colleagues, then studied cells in the area that contains the grid cells and the head direction cells and the speed cells and the border cells. But it turned out there is yet another type of cell that encodes space in a very different way. And these cells use a type of vector coding. That means distances and direction in relation to landmarks or objects in the environment. And that brings me then to, I have to remind you that until this experiment, essentially all of the experiments with these cells were done in rats and mice that walked in empty cages or empty boxes. There was really nothing in those environments. And that was deliberately done to keep things as simple as possible, which made it possible to find these grid cells. But still, it's not a very naturalistic environment. So all environments contain objects. So in this case, it could be this desk here, or it could be the door or the windows or whatever, which we use to find our way. We use the references to these places to find our way in the environment. And rats do the same. That's shown here. This is an experiment that was done quite a while ago, where Collett and his colleagues put, it wasn't rats, it was gerbils, but put them in a box like this, and they were walking here, and there were two objects here, two towers, and then at a location defined at a certain distance and direction from these towers, there was food that they had to dig to find. And they learned to dig at this place. That was easy. But then the experimenters moved the objects a little bit apart, and what they found then was that the animals started to dig in two places, either here or there. So those two places were defined by a certain distance and certain direction from each of the individual objects. So that then made several people construct models of how it might be in the brain, and suggested that maybe there are brains that in different ways encode distances and directions, or vectors, if you like, from prominent landmarks. But no such evidence was found. But here is data from a recent experiment where Øyvind and his colleagues in our lab recorded from cells and actually found that when you put an object into this environment, it's a circle environment in this case, and it's a mouse, you find that there are cells that respond to the location of the object. This is the object. The circle here is just this tower here of Lego, and it's moved to a different place here. But the cells don't fire at the object. They fire a certain distance away and a certain direction. And if you move the object to a new place, they still fire, in this case, on the north side, maybe 20, 30 centimeters away. And the same for this cell, also on the north side. Other cells might be on the south side or west side or whatever. So it seems like there's a separate category of cells. It's totally different from the grid cells that actually respond in a vector relationship to prominent landmarks in the environment. And then this just shows the distribution of preferred orientations. So you have essentially all orientations shown, and this shows the distribution of distances. So anything between very near the object, 0 centimeters away, to up to almost half a meter away, 45 centimeters in this case, which is almost half of the box. So then we further found that any type of object could really elicit this. So this is a variety of different prism-like objects. It could be cylinder-like objects, look quite different, but they all elicited firing on the side of the object. So in this case, for example, there are 3 of the objects in the box, and you can see that the cell is active on the southeast side, certain distance away in each of the cases. And you could even change the size of the objects. You say it's very small cylinder up to a very big cylinder, 30 centimeters wide, and you, in this case, still keep the firing field on the north side. Or you could change it to transform it from almost punctured object to a long wall gradually, and you can see that the cell fires along the wall as well. So many different stimuli may induce firing, and that then leads to the question, what is it actually about that object that causes this cell to fire? Because that almost brings us into philosophical question, what is an object and what is not an object? And since objects can be so different and still elicit this type of firing, what is the critical feature? So at least 2 categories of features that could induce the firing. It could be that those cells block the firing because they act like a boundary. They really prevent the animal from getting where it wants. It's sort of like a wall in the extreme case. Or it could be that it just has some sensory feature that is maybe high contrast or whatever. So the obvious experiment then is to just lift the object like this so that the mouse can't reach it. It's sort of hanging in the air or along the wall. And if you do that, what you find then is that the cells that have a firing field on the side here maintain their firing field even if it's hanging up and doesn't block the trajectory of the mouse any longer. So it is really not the blockade. It is actually the probably the sensory features of this object that causes the cell to fire. So it's probably related to having high contrast. So we're currently exploring that. All right. So now I have to adjust a little bit to time. So how many minutes more?
15 max.
15 max. Okay. So I want to briefly say something about one major difference between the place cells and between the hippocampal cells and all the entorhinal cells on the other side. Because very often, I would say, you can say that the hippocampal cells are high dimensional and the entorhinal cells are low dimensional. And what do I mean by that? Well, high dimensional means that you can have all kinds of combinations. So many, many ways they can sort of express position together. Whereas low dimensional means that it keeps the same relationships over and over again, regardless of where you are. So I'll give you examples to make that more specific. So this shows a recording from 2 different rooms. It's just an illustration, but it gives the point. So this is a set of grid cells, the green one and the red one and the blue one. And you can see that they have the relationship that I showed you earlier. But then if you now go from one room and record in another room, you will see that the relationship persists even in that room. So for example, the green one is always a little bit to the left of the blue one and a little bit below the red one. So the relationship is maintained. And that happens regardless of where you go. It is the same relationship. So it seems like it's just one map that is applied over and over again in every possible place. But then the question is, what about if you really go to the extreme and let the rats sleep? So does it then persist? So how can a map be recorded in sleep? Well, what you can do is first record them in the wake state, and you find a pair of cells that fire are active at the same location. So this shows a red grid cell and a black grid cell, very, very similar firing fields. Then you can plot the probability of firing in cell number 2 when cell number 1 is active. And this shows time on the x-axis. There should be time there. So it shows that when cell 1 is active, then the probability of cell 2 being active is very high. So they fire together. So this is in the active state. So this is kind of a consequence of that, no surprise. But if you then go to sleep, either slow wave sleep or REM sleep, you can see that these peaks persist. It means that when a pair of cells that are active in the wake state are also active in the sleep states. And opposite, if they are active in opposite places in the wake state when the rat is running in the box, and then have an opposite relationship, like here, this opposite relationship persists also in the sleep states. So that means that you actually have one map that even when you're sleeping, it's keeping its internal relationship. So it's really dependent on how the cells are connected together. It doesn't really care that much about the external environment. So these are maps that are sort of made by the entorhinal cortex itself, and not really dependent on the sensory inputs that come in. The sensory inputs are necessary to calibrate the system, but that's not really what creates it. And this is very, very different from the hippocampus. So what you see here is just very briefly an experiment where rats went from boxes in 11 different rooms, and in each room place cells were recorded, and then the place cells were put in a stack and then correlated between the different rooms. So you correlated the activity between room number 1 and room number 2, 3, 4, and 5, and so on, and then made a matrix. A color means the correlation between the rooms. And what you can see is that apart from diagonal, which of course is a correlation of a room with itself, then almost everywhere else is just in the blue area. That means a very, very low correlation, no similarity at all. Every possible room, the combination is totally different. And the only exceptions are if you do a repeated experiment in one of the rooms, which is where I indicated the stars there. So that means the hippocampus and the entorhinal cortex are very, very different in the sense that the grid cells and also the other cells of the entorhinal cortex is one single map that is applied over and over and over again, whereas the hippocampal map, which consists of place cells and many other, well, mostly place cells, they are totally different in every possible environment as a unique combination of cells. So what that means is that here you have one system that is actually quite suitable for measuring your position in space. It's a measurement system that can tell you in distances and directions where you are. And for that, you really want only one system. You don't want to have a new measurement system for every possible room. That would be extremely inefficient. But when it comes to the map in the hippocampus, that is actually a different type of map. That's a memory map where you create a unique map for every single environment. So for this room, for the hallway outside, or for your kitchen at home, or whatever, the maps are totally uncorrelated. And that's what you want. Keep them as different as possible, because then you don't mix them up in memory. So this is in retrospect, it looks like a very smart way of designing the brain. So with that, then now I have maximum 10 minutes left, right? So I'll, let's see, I'll speed up a little bit. But with this part, this is new data, unpublished, and I don't want to make too much out of it. But I do want to show that this is a work especially of Flavio Donato, where who was a postdoc in our lab, where we asked about time. How is this organized in time? Because we have all these maps, but these maps are sort of averages of what happens over time, but actually things develop in time. So how does this happen? And one way to do this is to have the mice run on a ball. It's a stationary ball, and then at the same time having a microscope over their head, so their head fixed, so that they just run in one environment during the experiment. And it's totally dark, so they see nothing, there's nothing happening, it's totally boring, but they tend to run because they like to run occasionally. And then Flavio and his collaborators recorded with a microscope, a 2-photon microscope that could then trace the activity of the cells, because the cells expressed had a fluorescent calcium image indicator, so that you can follow the calcium activity in the cells and see them turning on and off, which correlates very much with the action potential activity of the cells. And then you can get images of the cells. You usually get several hundred cells at the same time in entorhinal cortex by using a prism, that you put the prism in here so that you can image from the outside, from the back of the entorhinal cortex through this microscope. And then this then shows a typical result of activity. So here you have more than 200 cells organized from number 1 to number 200 plus, and this is time. And for each you see when they are active, so each dot is when they fire, when there's a calcium spike. And what you can see, it looks pretty disorganized. It's a total mess. So what you do when you have a total mess? Well, you can try to organize the data, see if there are patterns. And one of the techniques you can use is principal component analysis. You try to break down the data, see if there are clusters of activity according to which it's organized. So here, in this analysis, we organized the cells in time. So you define small blocks of times, and then organize the cells under each other, and ran this analysis, and then found at least 1, 2, 3 very strong components, and the other ones are much weaker, and then organized the cells according to how much they load on or correlate with the first component. So this is then we reorder the cells according to how much they load on this principal component. And this is what happens. So suddenly, if you see that there is actually structure in this data, they repeat over and over the same pattern over time, which you can then see by doing this advanced statistical analysis. So actually there is a pattern in the data. I will not have time really to go into what the pattern is like, but I'll show you here that this just is this a quite monotopic pattern that repeats over quite long time. So these cycles here are in this case up to 2-3 minutes long, and the network activity seems to drift through a sequence of many hundreds of cells, and then these sequences tend to repeat over and over again. And they are extra long when the animal is young. When it gets older and adult, they get shorter, but still are in the order of tens of seconds. So we are working on these sequences, try to understand what these trajectories are, but they seem to indicate, as I show here, that this must be consequences of how the network is wired up, because the activity sort of tends to follow the same trajectories through these complicated network over and over again. So I think this has a lot of consequences on how we think that activity is actually organized in this brain system and probably other cortical systems. So finally, I want to say a few words about time, because this suggests that there are mechanisms for actually arranging activity in time in the brain. But then, coming back to time, I haven't said yet that the hippocampus is also known, probably more known for its role in daily life memories. And I indicated indirectly by showing that it has the ability to store all these hundreds of thousands of different maps. But what also happens if you have a damage or a lesion to the hippocampus, as shown in the famous patient HM, then what you lose is your daily life memories, your ability to form and even remember new memories. So it's just like in dementias, that totally lost ability to take in new daily life type of episodic memories. And then that brings me to time, because episodic memories, memories of, let's say, how you got into this room an hour ago, that is organized in time. There's a beginning and there's a middle and there's an end. And we do remember that too. So how does that get into the equation? It's not only about space. So then looking for a time signature, then we, and that means especially Albert Sau, who was a PhD student in our lab, started to record in the other part of entorhinal cortex that I didn't talk much about, the lateral part, which is much, much less understood, and put the rats into many, many boxes over several hours, and they recorded them one after the other. And as time progressed, and then finally, after searching there for almost actually 8 years, because we couldn't make any sense of this, because the signal changed all the time, nothing was stable, total mess, then we found out that, well, actually it is recording the drift of time, the passage of time, and that's why it is changing. So there are cells that have activity that change with time. So this shows one example. So each block here is a black box, a white box, a white, black, black, and then you can see that in each box the activity ramps up, begins slow, goes up, up, up. Next box, begins slow, goes up, 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 and so on. So there are cells, if you break it down, you can see here the contribution of time and the position in the room and other factors like the color. But in this brain area, in the lateral entorhinal cortex, the cells primarily respond to the time that has passed. So I think this is the way that then from the lateral entorhinal cortex, this comes into the hippocampus. And from the medial entorhinal cortex, you get the spatial signals. They meet in the hippocampus and probably form the basis for episodic memories that contain both space and time. And I think with that, I won't really say much more about that in the interest of time, because it's 1 minute left. But let me, in the end, sum up by just saying that this is all, as you heard from the rector, collaborative work. There are lots of people, hundreds of people have been involved in this over a time of more than, or soon 20 years, actually. And I mentioned some of those who are involved in the specific experiments, and Britt Moser has been involved in all of the work, and then this is some of the early collaborators on the discoveries of grid cells, and a lot of people have, of course, paid for the whole business. So with that, I think it's time to thank you for your attention, and I'm happy to take any questions that you might have.
Thank you very much indeed, Edvard. So we do have 15 minutes for questions, and this is your chance. Just please go straight ahead.
Yes, so
there's a microphone, sir. Thank you.
So maybe this was obvious and I missed it, but with the experiment in the cage and the mouse and the plate cells, the grid cells, so we could see how when the mouse went to one corner of the cage, the one cell would fire, then another corner, a different cell, and so on. But it seems as though in the cage, there is a frame of reference because it has like an opening and everything. So if it was a perfectly symmetrical cage, would the same thing happen? I mean, how would the mouse know if he was in one corner or the other?
Yeah, that's a good question. Yeah, I think that did probably not come clearly enough across. So as I said, the combination of cells or the pattern of cells is very fixed and even happens during sleep. But nonetheless, in order for the same cell to fire, let's say a particular grid cells fire with a dot maybe in one corner and not in the other corner, for that to happen, they need to calibrate their maps with regard to the environment. So when a rat gets into the box for the first time, it sees everything that is around, and in order to prevent that it confuses the left side and the right side, or we actually deliberately put in what we call a cue card. It's just a sheet of paper on the wall that helps it to orient it so that the grid cells can then fire in the same location. If we hadn't done that, and we also tried to hide all the background cues, then if you have, as you say, a symmetrical environment, it could easily, the whole map would swap around, and that does happen sometimes if you don't have these cue cards, because the rat doesn't know what is east and west or anything.
Okay. More questions? Yes, please, at the back.
Good morning. Thank you very much for coming here and giving us such an amazing lecture. My question is, as far as I have understood, there are a different place cell for every single location we know. So does our brain have to develop new cells every time we visit a new place that we have never been there before? Because there are millions of possibilities.
Mm-hmm. No, there are, at every possible place, there is a unique combination of cells, but because the combinations that you can create among several hundred cells, they are almost endless. You can always make a new combination of cells be active. Then you don't really need to create new cells. But what you need to do is to connect different cells in a new way. And as I said, the opportunities for combinations are endless almost. But you need to connect them, and the way that is done, or thought to be done, is that you strengthen connections between cells, but making the synapses stronger or weaker, and you then store these patterns of connections so that next time the rat or mouse or human comes back, it can reactivate that same cell because it's stored in the memory. But when you then go to a new place, then you can create a new pattern, and that is stored. And if you ever come back, or even if you just try to remember it, you can reactivate because those cells are already then linked together. But as I said, you don't really need to create new cells. That would be a very inefficient way to do it.
More questions? Yes, please.
Well, first of all, thank you for congratulations for your presentation. It was very interesting. And if you allow me, I got two questions. Like, first of all, regarding these place cells, you have studied them in a two-dimensional surface. What would happen if we added a third dimension to the function of these cells? Would they behave differently? And the second thing, in an interview I read about you like yesterday and this your colleague Britt mentioned in the presentation she gave us last year, you both said that this experiment had potential applications for diseases like Alzheimer's disease. So I would like to ask you, what would be the next lines of investigation that should be followed in order to get to better knowledge? Or
yeah, so both of those questions relate to the presence of these cells in different species. So I mentioned very briefly that grid cells and place cells have also been recorded in bats. So those who did those experiments, initially they had these bats just crawl on a surface, but the next step was, of course, to have them flying. So they're flying in a big three-dimensional environment, a big room, and they try to record their position in that room in X, Y, and Z, and they find that, yes, place cells are three-dimensional in the bats, I mean, typically spherical, and head direction cells are also, so you have all directions, not only in the horizontal plane, but also in the vertical plane. And finally, now they have shown that grid cells are also three-dimensional. So then, how are they three-dimensional? That's still not clear, because there's several ways that you can actually pack information or pack clusters of activity in three-dimensional. At least there are two dominant ways, but it's at least clear that the distance between these clusters of activity in 3 dimensions is very, very similar between neighboring regions, which is similar to the hexagonal pattern in some way, although since they are flying, it's less accurate, so it's much more noise in the data. So yes, it does exist in 3 dimensions. And the question then is, to what extent is this developed in terrestrial animals that don't fly, like both rats and mice and humans? That we don't know, but we do know, of course, that we take, when we move around, we also take the vertical dimension into account. So most likely it's something similar also in humans. So, but that still remains to be shown. And when it comes to applications in humans, this is very strongly related to Alzheimer's disease. That was among the topics yesterday, because Alzheimer's disease very often tends to begin with cell death in the very same area that we recorded, in the entorhinal cortex, the first place where you get neurodegeneration. And that goes along with dysfunctions in finding your way. People tend to get lost as one of the first symptoms in Alzheimer's, and also memory problems. So it's exactly those functions that these brain areas support. So there is more and more research now, and in some of that is also involving our lab, although we still stay on the normal functions mostly, which tries to find out what is it that goes wrong in these cells. Why are they so vulnerable? Why are they so sensitive, and why does it begin there? That we don't know, but it may be an important part of the Alzheimer question.
Questions? Please.
Supposing the grid structure is only partially altered, could the person still have a partial sense of orientation within space, or would it just be completely lost?
Can you say again?
If the grid structure, the grid cells, are only partially altered, in the sense
only
that...
partially
altered.
Altered, yeah, okay, yes.
Would the person still be able to be oriented in the terms of space, or would it be a complete disorder?
Yeah, so I mean the question may relate to what you may consider a dream experiment. You really want to change the activity of the grid cells, or even turn them off, and then see what is the consequence of navigation. That is something we definitely would like to do, but we don't have a way at the moment to turn off selectively the grid cells. So if we inactivate, for example, the entorhinal cortex, all the other cells go as well. But that said, I mean, you can record grid cells under certain conditions where the grids sort of tend to be squeezed in different directions, which if you introduce barriers or objects into the environment, the grid patterns tend to use these objects or walls as references. And if you suddenly put one wall in the middle of the box, it will use that wall as one reference and then use the other external wall as one reference. And perhaps the two grid patterns, then one starting out from the middle wall and one from the external wall, don't match. So somewhere in the middle, it has to be resolved. And then you get a compromise grid that is not perfectly hexagonal any longer. You may then ask, does this affect their navigation skills? Well, in general sense, it does not. And the reason is that these rats and mice are probably have found a way to deal with this because they have to solve this problem in the natural environment. And what is matters most of all is that you do these adaptations in a coherent way, in a similar way across the many cells. And then the system has learned to deal with these distortions, which is necessary because you do get distortions in natural environments because of all the adjustments that have to be made because of objects and so on.
Yes, please.
Okay. My question is, do grid cells activate when you move the rat, or does the rat have to move itself to?
Ah, okay, passive movement. Yeah, that's an interesting question. So there have been many experiments done to try to find out whether active movement is necessary. So the typical way to do it is to, I show this ball, right, where rats or mice walk on a small ball that they move forward, but they actually don't move in space. And then they move in, we typically use a virtual environment, so it's like a cinema around the animal, and then you can either choose to move the environment according to the movement on the ball, so it looks like it's moving in real world, and you can, for example, move the world without the rat moving, so the ball doesn't move around, or you can sort of separate them. It turns out that movement is quite important, so they have to move by themselves. But there are also cells that can respond to just movement of the environment. That's what we call optic flow, that just you see the environment passing by, which also is felt like a kind of movement. So the brain can also use that. And even some cells respond very strongly to the objects, which I showed. There are other cells that respond to the objects. So, but yes, movement is very important, and it probably has to be active movement.
Thank you very much. Yeah, question. Maybe last question here. Okay. You okay?
Hi. Thank you very much. What is for you a single memory of being, for example, in a certain place at a certain moment? Is it a specific frequency of this stimuli, or of all these kind of cells? What is a memory?
What is a memory? Okay. A memory, a physiological view of memory would be a pattern of cells that is connected together by, I mean, a memory always contains many hundreds of cells, maybe thousands, that are active at the time of experience. And then those, a common view is that those cells that were active at the same time, their connections between them are strengthened, so that when you remember this sometime later as a memory, then that's because the connections stay strong between those cells, and you can just activate a small part of it and you'll get the whole network reactivated. The same network of cells that was actually active in the original situation when this happened. Because they have these strengthened connections, you can sort of reactivate it, and that's the memory. So that's the most common view of how memories are formed and maintained.
So there's no longer the idea of the grandmother cell?
No, definitely not. So memories are that. There was some time people thought about grandmother cells, as they called it, a sort of a cell that you had a cell for your grandmother and another cell for your aunt and another cell for your sister and so on. But it's not like that. It's all distributed among many, many cells, and it's all a result of collaborative activity in big networks of cells. They are not specialized cells for your grandmother or your uncle or whatever.
Did you have a question? Yeah, quick.
Well, this process you have told us, does require both sides of the brain working simultaneously, or if you have like one of the sides like completely destroyed or something like that, the other can take this function, or you have alterations or something like that?
Yeah, so I mean, these processes happen in both parts of the brain, and especially in the rat and the mouse, there doesn't seem to be very much specialization. In the human brain, there is some specialization, so there's more domination by language on the left side and by, for example, spatial relations on the right side. But they do communicate very strongly. But if you, for some reason, lose one hippocampus, for example, and keep the other, or one entorhinal cortex and keep the other can still keep going pretty well, actually. So you don't need the whole network really to perform quite well. And that's also why, I mean, during diseases, like in Alzheimer's, you can lose quite a lot of cells before it really matters, because you can do very well with just a small part of it. And that's also, if you lose one, you can still use the other for a long time.
Did you have a question? Last one.
So we've theorized about the idea of place and time, but if we talk about position, among all of us, we probably know someone who's got problems, difference in left and right, for example. Has it been theorized around that idea if these grid cells or place cells contribute to the idea of not only knowing if you're going straight forward, but also turning, how like you make that decision?
Yes, they are involved in the whole process of finding your way, whether it's straightforward or left or right. But what I haven't said is that this is part of an internal map of space, but this has to work in collaboration with other parts of the brain that make decisions and plans routes, and that doesn't only happen in the entorhinal cortex. For example, the frontal cortices are very important for making your plans about where you go and how you're going to get there, and maybe what you're going to do on the way. And so it is a whole brain network, but when with each area having their specialized functions, and they have to work together.
Thank you. I'm just going to allow myself one question from the perspective of people planning careers. You mentioned that Albert Sau, a PhD student, spent 8 years recording from lateral entorhinal cortex. Did he get his PhD?
Yes, he's one of the most successful students we have had. He's now at Stanford and is doing a fantastic postdoc, and I think you will definitely hear more from him. So, but it shows the persistence you need to have in this field. It is an extreme case, but it also tells something about him. He's totally dedicated, and he really wanted to find out what this lateral entorhinal cortex did, and he didn't give up. He did some other things on the side, though, I have to say that. But still, this was his big project, and in the end, after he had moved, actually, it was even after his PhD, then finally we got the breakthrough, and then by help of other people in our lab, the next generation, they helped recording the data that were necessary to really nail it. But it is certainly helps to be stubborn and highly motivated, but it doesn't have to be like that. It's very but that said, most projects in our lab last 5 years or 6. That is the average. So it's not something that happens overnight. It is a slow process, and the challenge is the PhD system in Norway, and probably the same here, doesn't really allow for 5-6 year projects. So that is always a problem.
Okay, thank you very much indeed. Thank you for a wonderful lecture, and thank you all for these extraordinarily good questions. It's been an excellent session.
Yeah, I agree. Very good questions. So thank you for all the preparation. As I said, when I came in, either you are very smart or you're prepared well, but then the director said, well, maybe both. So
thank you.
Okay.
to thank once more. Thank you for being here at work. And thanks for your advice, for showing us your research. And also for the last question, because I think that's a true problem. I mean, we are regulating so much. I mean, how the careers have to be, how long the grants have to last, that we have the risk of, yes, throwing away of the system very, very interesting people, and we have to be careful with it. So thank you very much.
Okay.
Okay.